Hi, God bless. Scott Stewart here. I want to talk about today the origin of life. In the beginning, God created the heavens and the earth. It's either true or it's not. I believe it is. Today, science tells us, or men who represent science or claim to, tell us that life happened accidentally, that it came about by mysterious circumstance. Funny thing, there's a lot of flaws with what they tell us. They keep on changing. Well, science changes. Well, what's true today is going to be true tomorrow. It might not still exist tomorrow, but what's true for today is truth. It exists. Facts exist. Things are what they are. Me thinking differently, you thinking differently, is not going to change anything. Truth is truth. Now, man has made great technological strides in understanding the world around them. Today, mankind is actually reshaping life. Scientists have created the first organism in history, to our knowledge, to have an entirely synthetic genome, the genetic sequence inside, and they've called it JCVI -SYN 1.0. You can look it up. And since then, scientists have been working to strip that organism down to its minimum genetic components. The super simple cell they created 5 years ago, dubbed JCVI-SYN 3.0, was perhaps too minimalist. The researchers have now added 19 genes back to the cell, including the 7 needed for normal cell division, to create the new variant, JCVI-SYN 3A. This variant has fewer than 500 genes. 492. To put that number in perspective, the E. coli bacteria that live in your gut have about 4,000 genes. And this is from the National Institute of Standards and Technology. So the scientists are saying that 500 genes have to be in this right sequence, or 492 genes, to have life. When they stripped it down to 283 or 483, there were problems with the life. It did not properly reproduce. It didn't excel. From Science Alert, we find when scientists synthetically eliminated a further 41% of mycoid's genes, leaving all but the most essential sequences, they created a free-living cell with the smallest genome of any organism grown in pure culture. Let's look at this real quick. In a pure culture, in a controlled culture, they were able to eliminate a lot of genes. Not in a natural occurring culture, but a pure culture, one that they designed to sustain life. They were able to get rid of some stuff. But they still have to provide the proper culture. I think of a virus. Viruses are thought to be more simple life forms, but they're actually more complex because not only do you need the stripped down genetics of the virus, but you also need the full genetics of the host organism. So it's not more simple after all, is it? Craig Ventner, the team lead of the team that created the first successful artificial genome, said, maybe you can't take a bunch of well-known genes, clamp them together, each one doing its own thing, and then when you tighten the last screw, suddenly, shazam, a new creature sparks into being. Instead of focusing on genes, maybe we should consider the whole operating system. Not the genes, but the genome, a functioning whole. And he probably doesn't realize, but he's admitting that the whole package has to come together simultaneously. That's exactly what he's saying. It wasn't a bunch of parts that came together mysteriously that created the first living cell. The whole package had to come together. The simplest natural cell has over 100 types of molecular structures and chemical processes, many found nowhere in nature outside of living things. Many others found nowhere together outside of living things that are in similar quantities together. You see? Some of these aren't even found at all outside of living cells. Each cell has to have a plasma membrane containing gateways called plasmodesmata or pores. These allow the hydration, feeding, and breathing. And these walls are comprised of various assortments of lipids, cellulose, etc. Each cell has a nucleus or a nucleoid consisting of DNA, enormously long strands of several complex proteins that need to be in proper formation to allow gestation, metabolism, growth, ingestion, expulsion, cell cohesion, organoid cohesion, maturity, and procreation of each cell simultaneously. You see? Each cell has endoplasmic reticulum, which are varied to synthesize various proteins, synthesize lipids, detoxify the toxins. Each of these have various functions, and there's variances in each of the reticulum. Ribosomes. These are organelles that attach to some reticulum to work with other factors in the cell to synthesize the various proteins. You see, it's a how did he say it? A whole operating system. Golgi, which are, they dispatch the proteins. They're like little buses or little trucks that carry the various proteins to the parts of the cell. Mitochondria. These are many types of organelles in each cell that digest various energy sources. Vesicles. And these are like the Golgi. They transport all these things to the proper destination. In addition, there's types of lysosomes. There's centrioles or their equivalent. There's microtubules and actin filaments which create a cytoskeleton in each cell. Each of these comprised of numerous varied molecules, proteins, fats, cellulose. All these factors, all of them, had to come together in the proper order, the result of a minimum 492 properly functioning genes. That's if it's a pure culture, simultaneously, all together. Do you realize that nothing in nature even comes close to that type of an environment that would bring all these things together simultaneously? It does not exist. There are no conditions found in nature that all these factors came together. If the 492 genes mysteriously came together, it would have to be in a functioning cell with functioning cell wall, cytoplasm, organelles, and nucleus. Over 100 different processes and molecular structures in the proper location and amounts to allow life. And that's just to allow it. What's going to kickstart it, right? All these conditions are met in only one location, an already living cell. And even this attempt that we talked about earlier at reverse engineering, that's based on placing an artificial genome in an already existing cell. They've already seen the blueprint for life in the genome, and they placed it in an already existing cell. In other words, life comes from life. Even this attempt at reverse engineering came from living beings, living intelligence. Life comes from life. And by looking at the miracle of the simplest living cell, we understand that life creates life. We had a creator. There is no other logical explanation. You want to talk about believing the science? That is the science. There is no other logical explanation. Romans 1:19 through 20. Because that which may be known of God is manifest in them, for God hath showed it unto them. For the invisible things of him from the creation of the world are clearly seen, being understood by the things that are made, even his eternal power and Godhead, so that they are without excuse. That's right, life came from life. The physical life came from the spiritual life of God. He created. He is our creator. And it behooves us, if we want to face reality, if we want to accept the science, to do our best, if at all possible, to find out who this God is, and to find out what it is, his relationship to us, what he intends. Amen? Amen.